about how how the letter areas on the sarcomere change, the A bands, H zones, and so on.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomere shortens. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
There are a lot more videos out there to be had on sarcomere function. Some of them will get into what the filaments are doing and the overall steps of getting the muscle contracted. So there's 4 major stages to muscle contraction. First, the muscle needs to be stimulated by a nerve. It doesn't contract without something telling it to. When you get involuntary contractions, your nerves are telling it to, but they might be going wrong and telling it when your brain didn't. Second is excitation-contraction coupling. It's basically how we get the message from the surface of the cell where the nerve delivers it down into the cell. Third, the filaments prepare. We need to expose those binding sites on actin. And finally, the cross-bridge cycle refers to the active contraction, where the muscle gets shorter. So, controlling contraction is through troponin and tropomyosin. If the actin was uncovered, the myosin would just grab and pull on it all the time. So we keep the tropomyosin covering it. When the nerve message comes through, we tell the troponin to move the tropomyosin. So to look at the message from the nerve, we need to look at one more piece of structure on this muscle. An area called the neuromuscular junction. So nerve, muscle come together. This green is the nerve. The cell is called a motor neuron. There's kind of a bowl in the sarcolemma that's called a motor end plate. The gap in between them is called a synapse. We'll see this again when we get to the nervous system. Any place that a nerve talks to another cell, whether it's a muscle, another nerve, or something else, is called a synapse. To get a message across that gap, we need something called a neurotransmitter. There's something like 30 different neurotransmitters used in our bodies, most of them within the brain. At the neuromuscular junction, there's one that's always used called acetylcholine. And that is the only neurotransmitter I'm going to ask you to remember the name of in this class, although I may mention some others as we go along. So here's the wider view. The motor neuron actually has a couple of branches that connect to multiple motor end plates, and that's just a way of reinforcing the message. We bring it at these 4 different spots. It's stronger than if we just bring it in one spot. On a closer view, you've got your motor neuron in yellow. The little circles inside it with the blue dots, those are vesicles storing the acetylcholine. Here's where the acetylcholine is released, and on the motor end plate there are receptors for it. Proteins in the surface that the acetylcholine can bind to, and that's how the muscle gets the message. So if we break this down into steps, the nerve stimulus. First, the neuron has a message travel along it. It's an electrical signal, we call it an action potential. we'll look more into how those work in chapter 10. Second, when it gets to the end of the motor neuron, the neuron releases the acetylcholine. Third, the acetylcholine binds to receptors on the motor end plate. So this is how a message gets from your brain to your muscle. Questions here? The next stage is excitation-contraction coupling. When the acetylcholine binds to the motor end plate, that starts another electrical signal or action potential on the muscle cell. That's going to travel along the sarcolemma and down inside through the t-tubules. Where the t-tubules contact the sarcoplasmic reticulum, they signal it, hey, we've been told to contract. The sarcoplasmic reticulum responds by releasing calcium, shown here as green dots, out into the sarcoplasm. So the calcium is how we tell the inside of the cell and the filaments that it's time to contract. So action potential goes in, T-tubules tell SR, SR releases calcium. Questions on excitation-contraction coupling? Filament preparation. This is sometimes lumped in with excitation-contraction coupling, but I find it easier to split it out. So once that calcium is released into the sarcoplasm, it's going to bind to troponin. Calcium ions are cations that have given up 2 electrons, so that makes them double positive. So you'll often see it abbreviated as Ca, either ++ or 2+. So if the troponin is the traffic cop who can move the roadblock, the calcium is the signal on the walkie-talkie telling him that it's time to open the road. So when the calcium binds, the troponin moves the tropomyosin. That exposes the binding sites on the actin, and it's all ready to go. Questions on filament preparation? Finally, the cross-bridge cycle. This is the game of tug-of-war itself. If we go back to the sarcomere illustration, but I'll do it smaller. It's not quite arranged the way a game of tug-of-war would be, because you've got 2 teams back to back pulling on ropes on each side, which are anchored to the Z line, instead of 1 rope and teams on each side. But we have to call the myosin the teams because they're the ones that do the pulling. There's 3 basic steps to it. If you think about how you play tug-of-war, we have our meters. Nope. So you're holding the rope. You're going to pull it close to you, and then before you can do anything else, you have to let go with one of your hands so you can reach out and grab it further along. And that's basically what the myosin is going to do. It reaches out and grabs the actin. Now if we just take one piece of myosin and a strip of actin, there's kind of a joint where the head meets the tail, where it can reach and pull at that point. So we're extending like this. Second, the myosin pulls the actin. That's bending the head, and it makes the actin move this way. And then third, the myosin releases the actin. Okay. And then that'll bring us back around to step one again, where after it's released this spot, it can reach out and grab another spot. So that's what the myosin is doing. We're going to add one more element to this, which is ATP, because you need energy to contract the muscle. What you need it for is to change the position of the myosin. The myosin already has an ATP on it when it reaches out to grab the actin. It has to split that ATP into ADP plus a separate phosphate in order to extend. Then, in order to pull, it releases the ADP and phosphate that are on it. So they're just out loose. And then to let go, it has to grab a new ATP. So I kind of think of it as if the myosin head is a hand, it can only hold so much. And it has to kind of drop off that phosphate to grab the actin. And then it doesn't have a good enough grip to pull, so it just drops the ADP, gets a better handle on the actin, and pulls. And then it goes, ooh, ATP, and lets go of the actin to grab the ATP. So here's a step-by-step. They've added in a little more detail, separated out. Now, at the top we have our filaments haven't prepared. The little pink spots are the binding sites on the actin. We put in our little calcium ions, troponin moves tropomyosin, and now we can do our cross-bridge cycle. So here's myosin grabs actin. This little yellow dot on it is ADP. It releases the ADP and pulls the actin. Let me see if I can get this a little lower. We can see there the change in angle of the heads. Then it releases the ADP, grabs ATP, and releases actin. And that brings it back to, it'll split the ATP and be able to reach out and grab the actin again. Here's an animation for that.
With calcium elevated around the myofibrils, myosin binding sites on actin are exposed, allowing interaction of the myosin cross bridges with actin. Release of the myosin cross bridge from actin requires binding of ATP. After ATP binds, it is split to ADP and inorganic phosphate. The energy derived from this reaction is utilized to cock the cross bridge in preparation for the power stroke. The power stroke is initiated when the myosin cross bridge binds to actin. The cycle begins anew when another ATP binds to myosin. At rest, most cross bridges are in the cocked configuration, prepared to interact with actin when the tropomyosin-troponin complex is moved away from myosin binding sites on actin by calcium ions.
Now, this model I've got here is one sarcomere. This outer piece is sarcoplasmic reticulum and t-tubules. So that's how the message gets in and the calcium gets released. And this is a little rickety, and the myosin heads don't do anything, but we can look at our sarcomere parts here. The Z line's in red, M line here, A band in blue, and then the H zone would be where it's not overlapping with the red. And I bands would be where we just have thin filaments. Okay. So when it contracts, the A bands stay the same size, but the actin overlaps more. That shortens the I bands and the sarcomeres overall. There's one other thing I want to do. I'm going to pause this and make sure we're lined up well, and then record just it.